Thanks for having me today. I'm just going to spend a little bit of time talking about some of the more recent work that I've been doing as part of my PhD thesis. So, the title of my talk is Exploring Disease Resistance Gene Candidates in Wild Rice. I'm a PhD candidate from coffee at the University of Queensland. And yeah, let's get started. So, just a little bit of background. We know that rice is a really important crop. It's a staple crop for over 3 billion people worldwide. It's one of the big three. Almost all of the rice that you would have eaten, regardless of what it looks like, has come from a single species, Oryza sativa. So 95% of our cultivated rice comes from Oryza sativa. Oryza sativa is divided into two subspecies, Indica and Japonica. And this species was domesticated in Asia around 9,000 years ago. There is one other species of rice that was domesticated in Africa slightly more recently, and that is Oryza glaberrima, and that was domesticated around 3,000 years ago, and that makes up the remaining around 5% of cultivated rice production worldwide. So just like all big crops, rice production is continually under threat from a range of different factors. Pests and diseases are one of the most significant causes of rice production losses annually. There's a number of pests and diseases that impact global rice production, but there are three major ones. The first one is blast disease. That's a fungal disease caused by the fungus Pericularia oryzae, also known as Magnaportha oryzae. And this disease is responsible for around 30% of global rice production losses annually, which has been equated to being enough to feed around 50 million people every year. So it's a very economically important disease of rice. The most significant pest of rice production is called the brown plant hopper, BPH. That's the insect pest Nilipovata lugens. And crop loss can be very high under high insect density, can be up to 100% crop loss. And the other problem with this is that BPH is the transmitter of several important viruses that cause disease in rice, like rice ragged stunt and rice grassy stunt viruses. And then finally, the most important bacterial disease of rice is bacterial blight or BB, and that's caused by Xanthomonas oryzae pathovar oryzae. And again, yield loss can be very high, up to 70%, when susceptible varieties are grown under disease-conducive conditions. So, um, the problem with disease resistance is that it can easily get broken down, especially when there's only single gene disease resistance in a cultivar. And in rice, we see disease resistance getting broken down every 5 to 7 years. So we continually need to find new sources of novel disease resistance to put into our rice cultivars. And a logical candidate or a logical option to look towards are the other non-domesticated wild Oryza species that exist and have evolved alongside a lot of these pests and pathogens. And so there are around 25, I think around 27 wild Oryza species that have been identified. They are very geographically, morphologically, and genetically diverse, found all over the world. This is not even an extensive list, but they can be broken up into genome groups based on how far away they or how distinct they are from cultivated rice. So first of all, we have the AA genome group. Cultivated rice is also in the AA genome group. So we have Oryza barthii and Oryza rufipogon. These are considered the putative ancestors of cultivated rice. But the AA genome group is also comprised of other wild rice species. This is just two of them, Oryza novara and Oryza meridionalis. Then slightly branching out a bit more, we have other diploid Oryza species, but these are part of different genome groups. So we have the BB Oryza punctata, the CC genome group, which is comprised of Echingeri, rhizomatis, and officinalis. And then we have a bit more distinct Oryza australiensis, the EE group in Australia, and the most genetically or evolutionary distinct Oryza brachyantha, part of the FF genome group. Um, and then finally, we have allotetraploids. So these have evolved from, I guess, like the merging of two diploid species. So we have the BBCC allotetraploids, um, including Oryza punctata again, which supposedly exists as both a diploid and a tetraploid. And then we also have the CCDD allotetraploids that exist in South America, South and Central America. Um, so a little bit about how disease resistance genes look in rice. So um, most disease resistance genes that we know of have come from ancient gene families that have been highly conserved. And we refer to these as resistance gene analogues. And resistance gene analogues can roughly be divided into two major classes. The first is the pathogen recognition receptors. And the key distinction with these is that they are embedded in the membrane and they possess an extracellular sensing domain for detecting pathogen molecules. The other class are the NLRs or the NBS LRRs, and they work intracellularly. They still have a sensing domain, but they work on the inside of the cells. The key thing to know about these is that both of these types of RGA, they are highly conserved and have highly conserved structures and motifs. And by that logic, because we know what these look like, we can go along a genomic region and identify candidate or potential new resistance gene analogues that look like the conventional gene analogues. So that brings me to um, a bit about what I'm doing for a couple of chapters in my PhD thesis. So I'm mostly working on the bioinformatics side of things. So in the last few years, there have been a number of very high quality reference genomes produced for most of these wild Oryza species. And so what I'm doing is collecting these high quality reference genomes and then going through them and searching them for resistance gene analog candidates using established tools that have been made for recognizing those conserved structural domains. And then I guess the end goal is that we identify candidates that can basically fast track the deployment of new disease resistance into crops. So once we have an idea of the resistance gene analogues across the species, we go and we run all of the genomes through the tools, we identify potential RGAs, we can then start asking some questions to try to get an idea of how they're distributed along the different genomes. So we can ask questions like, does RGA count increase as genome size increases? And we find that to an extent they do, but not necessarily all the time. So, for example, here we have our we have like a little cluster of genomes that have a higher NLR count relative to their genome size. And when we look at what these include the cultivated rices as well as those that are closely related to the cultivated rices. And if you think about it, this kind of makes sense because as we domesticate our cultivated species, we are actively selecting for traits that confer disease resistance. So it kind of makes sense that we are seeing more disease resistance genes in cultivated rices and those that are closely related to them. But we also see that the allotetraploids, they have very big genomes because remember, we're essentially doubling the genome size. And we see that similarly, they have a very high number of RGAs. Again, this makes sense because if you're essentially doubling the genome, you're essentially doubling the number of resistance genes that you see. So again, this kind of makes sense, but you can see some of the species like Oryza punctata relatively has a higher number of RGAs compared to its genome size. So these might also be interesting to look at to see how resistance gene analogues are distributed within these species. But then finally, we see some species that have a proportionally lower number of resistance gene analogues relative to their genome size. So, namely, Oryza australiensis and Mayeriana. These are two very large diploid genomes, but relatively, they have a lower number of RGAs. But despite this, we know that both of these species confer disease resistance against local pathogens and pests. So I guess we have to wonder how they're using the genes that they do have to confer these resistances, and maybe these are particularly interesting genomes that consist of gene variants that may confer broad spectrum or durable disease resistance. So these are some of the things that we can look at. We can also ask questions about how do RGAs compare with chromosome size? Are they evenly distributed along the chromosomes, or are they skewed on certain chromosomes? So I also had a look. These are just two examples, Australiensis and Brachyantha, but I had a look at the density of resistance gene analogues across chromosomes based on chromosome size. And you can see that it's, again, not straightforward. So, for example, chromosome 1 and chromosome 2 in Oryza australiensis, very similar of size, but very different number of resistance gene analogues. So maybe we want to look at chromosome 1 for potential hotspots of disease resistance that we could deploy into cultivated rice. And same goes for Oryza brachyantha. It does follow a general increasing linear trend, but we do see that in chromosome 11, we have a relatively shorter chromosome, but a very high number of RGAs. So maybe we want to look at chromosome 11 in Oryza brachyantha. So the other thing that I looked into, in addition to supplement the resistance gene analogues, was also looking at homologs to disease resistant genes in rice that we already know about. So we call these cloned disease resistance genes. And when I say cloned, I refer to genes that have been functionally and molecularly characterized in rice. So we know that they work. Um, and so when we look for homologs, we are looking for genes that are highly, highly similar to these known genes. And therefore, they are very likely and promising candidates that could hold novel disease resistance. So in rice, there are around 50 disease resistance genes that have been cloned, and all of these confer resistance to the big three, rice blast, bacterial blight, and BPH, and then a small handful of them confer resistance to viral diseases. So again, once we've once we've identified these homologs, we can then ask some questions. How do they differ along the genome groups? And we still see some similar patterns. So, for example, the cultivated rices have quite a high number of homologs. Again, this makes sense because they've all been functionally characterized in cultivated rice. But we can see that there are some differences in the tetraploids. So the number of homologs is not necessarily always higher in the tetraploids. Like that difference is not as pronounced compared to the resistance gene analogues. Although you do see with the allotetraploid Oryza punctata that does have a very high number of homologs and is much higher than its other BBCC genome counterparts. And then finally, similar to what we saw in the RGAs, the number of homologs generally decreases with genetic distance from cultivated rice. So we see the diploid, distinct, evolutionary distinct genome groups generally tend to have fewer clone disease resistance homologs. So we can also look more specifically at the proportion of genes conferring resistance to the different diseases in general. Um, and so what we found on the left here, we have the AA diploids that are more similar to cultivated rice, and on the right we have the non-AA wild diploids that are more distinct. I've kept cultivated rice in both of them just for comparisons. But what we can see, what we found was that the proportion of homologs associated with different diseases changes as you get more evolutionary distinct from cultivated rice. So the proportion of blast disease and brown plant hopper um, disease resistance gene homologs tends to decrease as you go more into the non-AA wild diploids. Whereas we saw viral um, homologs to viral disease resistance genes and bacterial blight disease resistance genes generally stay the same in terms of numbers. Um, and so again, we can visualize the distribution of homologs across the genomes, and this is chromosome 11 as an example. Remember, we saw chromosome 11 often harbors hotspots for disease resistance genes, and it's a known hotspot. So we looked at chromosome 11 to see how these genes are distributed. And we can see that from duplication analyses, there's a number of potential duplication events that have occurred, particularly in the AA genome groups. Some of them are quite interesting. So, for example, PB3, that confers resistance to panicle blast. There are only three cloned panicle blast genes. So this is an important type of gene that we'd want to be looking for. There were 15 PB3 homologs in the Australian Oryza rufipogon genome. And compared to all the other genomes, there were usually a maximum of around five PB3. So that's an interesting potential expansion of the PB3 gene that has occurred in the Australian Oryza rufipogon. And then again, we observe fewer of these duplications occurring in Oryza australiensis and Oryza mayeriana. Um, it is important to note that we did see a lot of single copies um, of homologs to each of the clone disease resistance genes, but we didn't see the same duplication that we saw in other species. I think that's probably likely due to the presence of retrotransposons. Both of these genomes have a lot of retrotransposons, which can copy and paste themselves along the genome, and these can disrupt uh, these can disrupt expansion events of other genes, but they can also create environments where duplication events can be more challenging. Something interesting to see was when I looked at the tetraploids, um, the the distribution of homologs across the subgenomes within the tetraploids was not uniform. So we found in both the BBCC and the CCDD allotetraploids, the CC genome tended to harbor a higher number of homologs, which was really interesting. And it also tended to harbor a number of, like, this wasn't just in terms of duplication, it also, when there were single copies of homologs, they tended to appear on the CC genome. So some genes were ubiquitous across both. For example, the XA21 gene that was found on both genome subgroups, but others, for example, the RGA4 and RGA5 gene pair, which have been really important historically in conferring resistance against rice blast disease, these were only found on the CC subgenome, which was interesting. One exception to this, though, was again Oryza punctata, the allotetraploid Oryza punctata again, which you can see on the bar graph on the bottom there. So for most of the tetraploids, the CC genome is relatively higher than the DD or the BB, but that's not the case with Oryza punctata. They're fairly even. And I think part of the reason for that is because Oryza punctata is a relatively new allotetraploid. It's occurred the most recently out of all of them. And so it has redundant disease resistance genes where we don't need, where duplication doesn't necessarily confer an advantage. So I think that's probably what's happened in this species. Um, And just briefly, I also looked at the specific NLR class. So remember, I said NLRs were one of the major classes of resistance gene analogues, but they're one of the most well-studied classes as well, not just in rice, but across all crop species. And so what we found was some interesting things when we compared the nucleotide binding site domains of the NLR genes and looked at, like, conducted some phylogenetic analyses. So we found that the motifs found in those conserved domains were like generally the same as what you'd expect in cultivated rice. But when we look at them on the phylogenetic tree, we can see some interesting things. So first of all, BPH14, that seemed to be the most earliest diverged or the most distinct out of all of the MBS containing NLR genes. We then saw some homologs had a single truncated NBS domain, which means that it, which like suggests that maybe these are non-functional, and these homologs were largely confined to certain genes such as PB2 or PI54. We see RLS1, which is a unique NBS containing gene that has armadillo repeats. We see that it's put on the same clade as PIT and PB3, two blast resistance genes. But RLS1 confers resistance against bacterial blight. And you can see that it's very, the homologs to RLS1 the branch lengths are very short and it's very much like distinct from other homologs. We can see here that the homologs to BPH18 and PIB, they have two MBS domains, but one of them is intact and the other one is truncated. Pick2 was interesting because the homologs to Pick2 didn't have intact MBS domains. Instead, it had one truncated domain coinciding with the nucleotide binding site domain and one truncated domain coinciding with the ARC domain. So together they made a full domain, but they were fragmented within the gene itself. And then finally, you see this large clade of genes corresponding to PI36, RYMV3, and LS1. And these were interesting because they had multiple intact NBS domains within their genes. So what this does, I'm not exactly sure, but it was interesting to see. And so finally, one other thing that we can do when we're looking at the homologs is we can gain a better understanding of how disease resistance works, or we can help shape a story of disease resistance genes where we don't really know the full story of how they function or what they're useful for. So an example of this is PETA and PETA2, which I think is a really interesting story. So PETA and PETA2 were originally identified together as a gene pair on chromosome 12, and it was believed that they worked together to confer blast resistance. So PETA is a conventional NLR. You can see on the top right here, it has an N-terminal, an NB-ARC domain, and then a leucine-rich repeat at the end. So that's a conventional NLR. And Peter was cloned in 2000. Peter 2 was more difficult to identify and clone because it's an unconventional resistance gene. And instead of having an NBS domain and an LRR domain, it is comprised primarily of these armadillo or arm repeats. And so Peter 2 was cloned in 2018. And originally, it was thought that Peter 2 was responsible for recognizing the pathogen, whilst Peter 1 was responsible for eliciting a response. Um, but then recently, in a recent study, it was found that they actually don't really necessarily work together at all, and that Peter 2 can function completely independently of Peter. And so I guess now there's a lot more questions that have arisen about these two genes and about how they work. And so it was interesting to see how they were distributed along the wild genomes um, because it kind of can shape how they are or aren't dependent on each other. So, for example, Peter 2 was only found on the AA and CC genomes within diploids and tetraploids, and it wasn't found on any other genome group. So you can see here highlighted in red Peter 2 is found in the AA genome group and the CC genome group, the diploids. You can see that Peter and Peter 2 are paired together, and you can see that they are found both on chromosome 12, but they're only found together on the AA and CC genome groups. And when you look in the tetraploids, you can see that Peter 2 is only found on the CC subgenome of the tetraploid group. And what's interesting is that in the BBCC allotetraploids, Peter and Peter 2 have been retained on different chromosomes. So Peter has been maintained on the B genome, and Peter 2 has been maintained on the C genome, which is kind of interesting that they they have been separated um, or been retained differently on those genome groups. So, and I guess when we look at the alignment of the arms domains in Peter 2, it's also interesting to see that there is clear variation between these these genome groups. So you can see here the CC clade um, is clearly distinct from the AA clade. And you can see several variations that occur. But even within the AA and CC branches, you can see that the subclades also have distinct differences. So several of the AA genome groups, including Oryza sativa subspecies indica, was on a separate clade to Oryza sativa japonica, and you can see that there are several variations along the alignment that demonstrate that there are distinctions between these homologs. And then also in the CCDD tetraploids, you can see that there are these differences that occur that don't occur in the other CC genomes. In Oryza meridionalis, we also have this insert here that's not seen in the others. And looking at what this insert actually was, this was found to be an additional duplication of an arm repeat. So whether this impacts disease resistance or not, we don't know, but that's something interesting to look at. And then finally, the C terminal of all of these genes was highly variable. And we know that disease resistance and disease susceptibility does vary with variations on the C terminal. So this is also possibly an area that we want to look for novel disease resistance in the Peter 2 gene homologs. So I guess quickly to summarize, the wild Oryza are extremely diverse, and because of this, they offer novel sources of disease resistance to local pathogens in cultivated rice. And in silico genomic analyses of potential disease resistance genes are important because they can help shape our understanding of how disease resistance has evolved within the genome, and it can also um, quicken how we identify obvious candidates for future rice breeding, so we can incorporate novel disease resistance into our crops more rapidly. Okay. Thank you so much for listening, and I welcome any questions if there are any.